Hi, David. How's it going?
Going very well. Looking forward to speaking with you again.
Yes, hello. And we are in part three now of our classification of viruses, and this time you're going to explain about DNA viruses.
Yes, and I wanted to start by discussing the fact that this isn't just a matter of taxonomy, going through the different types, because understanding the nature of the different viruses and the replication actually explains quite a bit about their lifestyles, how they interact with their hosts, how they interact with humans. So I think that's very important to understand. So these viruses that we're going to talk about are DNA viruses. And DNA viruses have as their genetic material, which they carry in the viral particle, DNA rather than RNA. And as I've mentioned before, DNA is a much more stable molecule than is RNA. And it is also the material that acts as the genetic material in the nucleus of cells such as mammals, such as ourselves. So in that sense, the storage where the genetic material, this genetic information is stored is similar in DNA viruses to what it is in the host, such as ourselves. And that separates them from the RNA viruses. So if we could talk just a minute about the different ways that DNA can potentially be included in the virus particle. So everybody's familiar with the fact that DNA, as the genetic material in a animal cell, for example, consists of two strands that wrap around one another. I'm not going to attempt to show them wrapping around each other, but it's a double-stranded. It forms what's called the double helix, right? Now, and that's the form that it takes in cells. However, some viruses, it does look like this. And just like a chromosome in a animal cell, it is a double-stranded molecule that is linear. It has a beginning and an end. In bacteria, the chromosomes tend to be still double-stranded, but they are in fact circular. So they are circular DNAs. And we have viruses that are like this, that have a double-stranded circular genome. So we have ones that are double-stranded and linear. We have ones that are double-stranded and circular. We have other ones that are single-stranded DNA viruses and circular. We have other ones that are single-stranded and not circular. So in every single case, when we're replicating the DNA, we always have to go through a form that is double-stranded, whether it's linear or it is circular, whether it's starts off as single-stranded in the virus particle or double-stranded in the virus particle. But in the cell, during the replication process, we always have to have some sort of form that is double-stranded. That's how the replication takes place. Now, the implications of the fact that the virus is made, its genetic material is DNA rather than RNA has a number of important implications. The first is that the genome, the genetic material of the virus, is much more stable in the form of DNA versus RNA. So that's the general principle. As I've mentioned before, RNA was the original source of genetic information. RNA was how life evolved first, was in terms of RNA. DNA was a storage form of nucleic acid, which came later. But the advantage of it as a storage medium is that it's much more stable. And what do I mean by more stable? Well, it's chemically more stable, but it's also more stable to genetic mutation. So it's, estimates 1,000, 10,000 times as more stable. It is less likely to have mutations than is RNA, for example, in an RNA virus. That's speaking in general. We're going to talk about the coronaviruses, which are a special case later on. But because it's more stable, that means that the DNA genome, the amount of material that can be present in a DNA virus, can be much greater than that present in an RNA virus. Because it's more stable, it can be transmitted more stably. It can be larger because when you think about the size of the genome and how many mutations there are, if there are lots of mutations in the small genome, it will be able to change fast. But it's likely that most of those mutations will be deleterious, and that'll be harmful to the transmission of the virus. So RNA viruses tend to be much more compact. DNA viruses have the luxury, because of the lower mutation rate, to be considerably larger. Well, what does that achieve for them, that they're larger? Well, one of the things they can do is, for example, the class of viruses called the herpes viruses, which includes Epstein-Barr virus, the herpes virus that one knows about, has heard about, varicella zoster that causes chicken pox, and also when it reactivates, it causes
shingles, right?
Shingles causes shingles. Those have many, many genes that they've acquired from the host, or that are modifications of genes acquired from the host, and those allow it to manipulate the immune system very effectively. And what does that achieve? Well, one of the things that allows is that those viruses, DNA viruses, can persist in their host for long periods of time. They can persist even for the lifespan of the organism. And they will be partially seen as being just like any other DNA. They frequently will be in the nucleus. They sometimes have a regimen where they will replicate only when the DNA in the nucleus replicates, and they're partially dependent upon the replication machinery that's present in the nucleus. It's certain metabolites and so on that are necessary. Sometimes certain proteins that are necessary. So they are in a that just allows them to adopt a very different lifestyle from that of the RNA viruses, and that has major implications for how and whether they cause disease, how long they persist, and how they evolve.
Just explain a little bit about why it is. What's the necessary connection between stability and persistence? Well, I mean,
sure.
I assume that the idea is that they are less susceptible to the immune system's attacks.
That the large size allows them to include proteins that manipulate the immune system and also potentially manipulate the cell cycle so that it permits them to be replicated. The RNA viruses, their materials, they don't require going through the cell cycle in order to create the materials that they need for replication, whereas the DNA viruses frequently do. But because of their larger size and their larger stability, they can encode proteins which will manipulate both the immune system and the cell cycle, and it's the ability to replicate the genome of the DNA virus, as well as potentially affect the replication of the host cell genome as well.
I see. So for some of these DNA viruses, the only way to get rid of them would be to find by way of pharmaceuticals or something.
Yes. So, you could potentially have a vaccine, right, that when you're initially exposed to them protects you against them. But when we're talking about these immune escape functions that it has, it's mostly for the earlier steps of the immune response. But when you generate, and we'll talk about the immune system in more depth and how it relates to viruses later on, but when you have the adaptive, the memory parts, it doesn't help you as much if the virus is permanently in the cell, right? That because you are most of this, if you are manipulating the cell so that the cell doesn't recognize that it's infected, that's one of the things that, for example, herpes viruses do, and they're persisting inside the cell, not in an extracellular form, so they won't be recognized by antibodies. The memory response just isn't as effective as it would be when we're talking about an RNA virus, which has to constantly be undergoing an extracellular form in order to be able to persist and propagate itself.
I see. When in the first podcast, you explained that the definition of a virus was a obligatory intracellular, right?
Molecular.
Yes,
obligate intracellular molecular parasite, an OIMP.
But now you're saying that DNA viruses are not intracellular?
They are intracellular, yes.
Oh.
That's what I'm saying. That's precisely what they are. They persist as an intracellular parasite, whereas RNA viruses tend to be hit and run. They tend to infect and then leave the cell as quickly as they can. Whereas DNA viruses, again, I'm generalizing here. Not necessarily all DNA viruses persist, and there are some RNA viruses that seem to persist in cells, but as a general principle, that's an important distinction to make. So they are intracellular, and they're actually many of them are actually maintained in the nucleus of animal cells.
So they're intracellular rather than intercellular, you mean?
That's great. Intracellular, inside.
Intracellular.
Right.
Okay.
Intracellular.
Right.
And so most DNA viruses now, as I mentioned before, influenza virus, the RNAs actually have to make it into the nucleus. So that's something different. And the pox viruses are a very different group of viruses. And as I said, RNA viruses tend to replicate in the cytoplasm. DNA viruses tend to replicate in the nucleus, but pox viruses replicate in the cytoplasm, and they make their own little environment there in the cytoplasm. They don't actually go into the nucleus. And I sometimes refer to pox viruses, which are a class of DNA viruses, as a cell manqué, which means that we've talked about the origin of viruses. Well, pox viruses, maybe, some people think this, some people think other things, but it may be that they are a sort of degraded form of a cell that just became a virus. But they whether or not that's their origin, they certainly have characteristics which suggest that they really do a good part of metabolism inside these little factories that they create for themselves in the cell. So they're not cells. I wouldn't want to call them cells, and they can only replicate inside a cell. But they have many of the characteristics that we would otherwise associate with cellular metabolism.
Okay, David, I think that's very interesting, and let's end that podcast there, and we'll come back and speak about retroviruses.
Look forward to that next time. Thanks a lot.
Thanks, Dave.